Hello, everyone, and thank you for joining us for today's webinar, Explore Millions of Clinico-Genomic Data Points from the UK Biobank. I am your moderator, Brenton Pyle, and I am excited to introduce our speaker, George Asimonos, DNAnexus Chief Technology Officer. George has been with DNAnexus since its inception and has been involved in the design and implementation of the DNAnexus product line and its application in key accounts around the world. George is currently heading up strategic projects using DNAnexus technology to craft novel experiences that transcend traditional genomic boundaries, including the creation of the UK Biobank Cohort browser being demonstrated today. If you have any questions during the webinar, please submit them via the chat or Q&A icons at the bottom of your Zoom window, and we'll answer them at the end of the presentation. The webinar is also being recorded. This recording will be sent out in the next couple days, so make sure to keep an eye on your inbox. Now, without further ado, I'll hand it off to George to kick it off.
Thank you, Brenton, and thank you everybody for joining us and for giving me this opportunity to present to you today our technology and how we've used it in the context of UK Biobank. I understand that some of you might not be familiar with the UK Biobank study, so I wanted to spend a few slides telling you some of the important information to give you some background and context around this project. It all started back in 2010 when people in the UK woke up to this letter in the mail inviting them to join this exciting national medical research project, asked people from the ages of 40 to 69 to enroll and to help study the prevention and treatment of several serious diseases. They were looking for 500,000 people total to join the project and to go into an assessment center of sorts where they would be measured and they would be asked all sorts of questions and they would contribute their blood to a biobank, the UK Biobank. This actually was a very successful project, and we have a lot of resources that the biobank has put in the public domain to understand exactly what happened. In fact, they even have this floor plan of what the assessment center was like. So someone would enter from the left and go through all of these stations, and they even have photos of the things that people did in this assessment. For example, there was a touchscreen interview for the participants to answer some questions and to help provide self-reported information, mostly about diseases and health information. So this is an example of this screenshot of the touchscreen display where they were asked, has a doctor ever told you that you have diabetes? And they were presented with 4 different answers, and they could touch the answer they felt was appropriate for their case and then go to the next question and so on and so forth. Obviously, not all the questions were related to health. In fact, here's another example of what type of cereal do you mainly eat with all the different UK-derived cereal categories. And then they were subject to all sorts of other things. For example, they had to play little games that measure their cognitive function. They had to go through a verbal interview with a nurse, where the nurse asked them a little bit more intricate questions about their health, their medications, and their diseases that are beyond what the touchscreen display would offer you, so they could codify the answers in a more intricate and detailed manner. And then they went through all sorts of measurements, like this is how waist circumference would be measured. This is an eye exam for eye refractometry. This is a fitness test where I think they had a cardiogram at the same time while also being on a bike and so on and so forth. So this is a great collection of measurements, a bit of a physical, if you may, that all these people undertook, 500,000 people from the UK. And further to that, their medical records were linked to this resource. And this is something that the UK can do more easily than perhaps anybody else, in the sense that the health system allows a very small number of providers to concentrate the information and link it to the UK Biobank. So information such as death data, cancer registries, and hospitalizations through these providers that you see here were also linked and provided to the biobank. So there's now information not just from what these people mentioned in their touchscreen display portion, their measurements and their verbal interviews, but also their hospital diagnostic codes, their reason and date of death, and so on and so forth. And of course, this is a biobank after all, so blood and other samples were retrieved for assays to be run in the future. And up until recently, there was no next generation sequencing information until one of our customers, Regeneron Genetic Center, decided to undertake a large exome sequencing effort and slowly get all 500,000 samples from the UK Biobank, and with the help of a large group of pharmaceutical companies fund and this process and do it. So they receive samples from the biobank, they sequence them in their lab in Tarrytown, and they produce exome sequencing results such as nucleotide level variants and other things. And they are contributed back to the biobank, which after a period of exclusivity makes this publicly available. And very recently, you may have heard that the first 50,000 exomes have become publicly available. And those alone contribute to a wealth of data. In fact, we promised you millions of clinical-genomic data points, and here's why. Because in 50,000 subjects, there's approximately 5 million genomic variants in their exome coordinates. So that alone contributes 250 billion genotypes. And then the phenotypic portion, all these measurements and all these questions that people were asked are in the order of 3,500 or so phenotypic fields that you can extract from the biobank. Therefore, they contribute several hundreds of millions of cells. And it would be intriguing if there was a way to quickly interrogate that and get some insight out of that data. And this is where DNAnexus Apollo, the technology we use for structured data and quick investigation, will be showcased today. In fact, I'll show you just one little piece of that solution, which is the visual analytics portion of it, the Cohort browser for UK Biobank, to give you an idea of how our technology powers large-scale genome-pheno investigations. So let me pause here and move to a real demo of the browser, which I can't quite show you the real UK Biobank data. So we have created a version that simulates data as it looks in reality on 50,000 individuals. And I have deliberately here cleared my view in order to build it from scratch and to show you how this is a very user-centric approach where you can get to choose what data to work with. So here you see at the top, it says 50,000 selected individuals, because I haven't started actually doing any investigation or filtering. And you will notice I have two main portions. The dashboard at the top allows me to look at phenotypic attributes, and I haven't selected anything. I'm deliberately keeping it empty for the time being. And down below, we have the genomic portion that shows all the genomic variants in the cohort. So I will focus for the time being on the phenome, and we'll get into the genome slightly later. So if we are going to be looking at stuff, we understand that the biobank has generated all this information. We're not quite perhaps sure what we want to work with. So we'll start looking at what data is available and add them here in our view. And you do that by clicking Add Tile, and you get to a list of all the phenotypic attributes. Remember those 3,000 plus different measurements that have been thankfully arranged into a folder hierarchy by the UK Biobank. And those that are approved researchers of the UK Biobank and have worked with the data will recognize this folder hierarchy right away. For example, if you're interested in the gender, that's a baseline population characteristic. So you go in this part of the folder hierarchy and then look at the sex. And you see a little preview showing you in this example synthetic data of 50,000 people, what's the distribution of male and female. And if that's something of interest to you to work with, you can add that to your browser in the back, and then you continue being in this UI so that you can choose other things that you can work with. For example, the age of the participants at recruitment. This is an integer field, so you see a distribution here as a preview, and then you can click Add as Tile and add that as well. Those that are not as familiar with this hierarchy might want to leverage the search capabilities to find something of interest. So if you're not sure where something might be, and especially because this is organized by how something was collected. Was it during the touchscreen interview? Was it during the verbal interview? Was it a physical measurement and whatnot? So you might be better off searching for something. For example, let's say we're interested in the height. So you see here, we have all these results. For example, during the physical measurement in anthropometry, there was a standing height, a seated height, sitting height, and whatnot. So this is an interesting aspect of the UK Biobank. There's a lot of different data points that are sounding a little similar. So it's a lot of work to identify what is the right thing for you to work with, which is why this UI of DNAnexus Apollo offers a lot of metadata to help you understand what's in your study. And this is also a characteristic of other data sets. You could be envisioning the same UI on your clinical trial data or on your own studies outside of UK Biobank, where you're not sure exactly what's available in your study and what you can use. So when you click on something, we have all of this information on the right-hand side. How many participants have a value for that field in this data set? What's the value type? What are the units? As well as link outs to external resources that document how that field came to be. For example, here in the standing height, if we were to click this link, we'll go to the UK Biobank showcase, which has information as to how the standing height was measured. In fact, there's even photos here for how it was measured. But if we look at height, for example, we see that it's been only measured for 2,000 people. And if we go to the UK Biobank showcase, we notice that this is something that's part of a calibration for a DEXA scan, a quick MRI device that many people haven't had the chance to undergo this experiment with the UK Biobank. Therefore, this has been conducted for very few people. So you see this height is only available for 2,000 people versus the standing height is available for 49,000 people. So maybe that's the one that you're interested in, and then you can add that as a tile. You continue here to look at things of interest and adding them. For example, if we look at the disease like diabetes, we'll notice that there are many sources of information here. For example, in the touchscreen portion of the assessment, there was a question, have you been diagnosed by a doctor for diabetes? And it was a bit of a yes, no, I don't know, or I prefer not to answer. But there's also, during the verbal interview, a classification of diseases that the UK Biobank created based on people's answers, and diabetes was an answer here, and that also shows in the search results. So this doesn't search just the names of the fields, searches also the dictionaries to allow you to find the information no matter where it might be hidden. So maybe that's something we're interested in, or if we look at some of the hospital records, there are diagnostic codes from the International Classification of Diseases, this ICD-10, which is a hierarchy of diagnostic codes. So every time you get hospitalized for something, there's a diagnosis that comes in a code, and these codes are organized in a hierarchy called ICD-10, and there's indeed a portion of that hierarchy for diabetes. In any case, there's a lot of different information here. For example, there's medications that people take and that they mention during a verbal interview. You can literally spend hours here investigating your favorite thing and adding it into the browser. When I first showed that to the pharma consortium, someone said, oh, can you search for miserableness? And I wasn't sure we would find anything, but sure enough, there was a question about, do you ever feel miserable for no reason? And it was surprising that almost half of the people in the UK Biobank had said yes. So expect that many different things might appear here. So it's interesting that you have so much power to search, to understand the metadata, and to add them. And lastly, I want to mention some of these assessments were conducted more than once. There was, in fact, this initial visit where everyone was asked these questions and did this measurement. There was also a first repeat assessment where people were asked to come back to redo it at a later point, but not everyone has been asked to come back. And then an even smaller group has been asked to come for an imaging appointment, typically an MRI, and they have the opportunity to ask those questions a third time. So it's usually these three distinct milestones, and the system allows you to work with each milestone separately. So if you look at the initial assessment visit, 49,000 people answered that question of the 50,000, but if we look at the first repeat assessment, there's only 2,000. So this drops as you make more progress. So it's not necessarily something that you can track longitudinally in a meaningful manner until more people have conducted this. But it's along the lines of what you would perhaps use if you were to load your clinical trial data, and then you had specific milestones for specific visits, and then you could envision them appearing here as you enter them into the system. So we've added all of these things, and now they appear here as tiles in my dashboard. So these are things that I can rearrange, I can move around, and I can resize whichever way I want. So maybe I want to have this diagnostic code be larger, and I can do it like this. So it helps you build the dashboard whichever way you like. You can add, remove, resize, move around, and so on and so forth. And you can always change that as you make progress, right? You can go back and add tiles at any point, remove tiles, and so on and so forth. So this shows you the distribution of all these values in your cohort. Now we're looking at the whole cohort. So for things that are a simple categorical field, like the gender is female or male, it shows us this row chart of sorts where the values appear as horizontal bars. And that's the same with a question such as, have you been diagnosed by a doctor for diabetes? All the possible values, this is a single choice question, are shown again as bars. And you'll notice that the order in which they appear is the same than the order in which people were asked this question, the natural order of sorts. So when you bring data into DNAnexus Apollo, you have the opportunity to bring dictionaries, but you also have the opportunity to say what is the order in which those dictionaries were shown to the user, or were put in the case report form, or were interrogated, so that you understand the reason why we show them in the exact same order. If the values were way too many, and this is the case for medication, it's almost like a drop down with a gazillion choices, then the order doesn't necessarily make much sense in terms of how they were presented. It was too many anyway, so we show them in order of frequency in the cohort. So in the medications down here, you'll see there's really many medications, and they appear in the order in which the frequency in this current cohort. And then if something is a numerical value like age or height, we compute distributions and show them like these histograms here. Some fields are a little bit more intricate, like these cancer, self-reported non-cancer illness code on the lower left, or these hospital diagnostic codes. They have been categorized into a hierarchy. In fact, here you see, for example, diabetes is this E10 to E14 part of chapter 4, and it has many different versions, whether it's insulin dependent or non-insulin dependent, and whether it's with ketoacidosis, renal complications, and this and that. So usually when you go to the hospital, you get one of or more of these codes assigned, typically the leaf nodes, but not always. And then our technology computes automatically counts for every internal node without double counting. So these 2,501 means that many people have had a diagnosis under E11. So this could be anywhere in here. And if someone has both with coma and with renal complications, has both codes, we don't double count it as we move up. So the technology is sort of smart enough to understand where you are in the tree if you are a patient, count you only once as you walk up the tree, and so on and so forth. Anyway, so this is the main visual for your cohort, and then you would want to start filtering it. So let's say you're interested in females that are above 60 years old. So you can brush this histogram, you can click on female, and then you can refresh the dashboard. So what's happening here at the top, you see we are down to 11,761 people from the 50,000 that we started with in the cohort. And then the filters appear here in cohort filters. You can mix and match between pheno and geno filters. Right now I'm applying only pheno filters. So I'm looking only at females that are above 60. Those filters are shown here, and then everything else is a distribution in your cohort. So here's the height of these women above 60. Here's their answer to whether they've been diagnosed for diabetes, their hospital codes, their medication codes, and so on and so forth. And as I start to apply more filters, I will drill down even further. In fact, I see these numbers, therefore I know how much my cohort will shrink if I apply more filters. For example, if we want the women that have replied yes, we know these are 556. So if I click on yes, and then I refresh, I know that I will be going down to 556 people in my cohort. So by showing you the distribution of any given field before you click it, you understand how many exist, and then you understand what you will go down to if you start filtering your cohort even further. Sometimes, however, you have things that could be combined. For example, here in these treatment and medications, medications are not necessarily mutually exclusive. So I could say I want everyone who is taking ibuprofen, and these are 58 people, but that's not the only drug that they're getting. So if I click ibuprofen and I refresh the dashboard, I will indeed go down to 58, but you'll notice that the numbers here in this box that shows the treatment and medication codes will change and will have two columns. And that's a question I get very often. What do these two columns mean? What's the cohort versus the cross filter? And it's really the following concept. Here we've selected for those that take ibuprofen. So the first column shows you what's happening in your current cohort. And because we're filtering for people with ibuprofen, obviously ibuprofen will be at the top because all 58 people in the cohort take ibuprofen. But that's not the only thing they take, right? So 8 of them also take aspirin, and 6 of them also take paracetamol, and so on and so forth. So here's what's happening in our cohort within women who are above 60, who have been diagnosed with diabetes, and who take ibuprofen. That's the other drugs, that's all the drugs they're taking. The second column, however, shows you what drugs people take if you momentarily ignore the ibuprofen filter. So this thing on the right shows you the drugs that women who are above 60 and diagnosed with diabetes take. If we were momentarily to ignore the fact that we filtered within this tile for ibuprofen. So the left is what's going in your cohort, and the right is what's going in your cohort if you were to ignore this ibuprofen thing. And sort of like a baseline for all medications in your cohort. And the reason why we do that is because you now have the freedom to choose how to go from here, whether you need to go down or you need to go up. For example, if you say, I want ibuprofen and paracetamol, you're gonna go down to 6 people because you have 58 people who take ibuprofen and 6 of those take paracetamol. If you wanna say, I want people who take ibuprofen or paracetamol, then you will be going up and you'll be adding up to 88 people minus any duplicates, because that's how many women above 60 who have been diagnosed with diabetes take paracetamol without regards to ibuprofen. So the left is how much you'll shrink if you say and something else. The right is how much you will expand if you say or something else. And you can actually click on this and choose whether you wanna do an and or. match any of these medications or match all of these medications. And as you decide what to do, these numbers always change. So here, if I refresh, now I've applied a ton of filters, right? So I'm looking at women who are above 60, who've been diagnosed with diabetes, who take both ibuprofen and paracetamol, and I know these are 6. So as we drill down, the numbers here become smaller, the distributions become thinner, and so on and so forth, because I'm down to a very small cohort. The same concept of those two numbers applies everywhere. So you see, for example, we're looking at these 6 women, but and there's no one who is male in our cohort, so this is 0. But there is a faint bar here that's actually twice as large as the female, and it says of 12, because this again creates a background distribution of this field in your cohort. So it says, you know, if I disregard momentarily the gender in your filters, and I look at anyone who's above 60, has been diagnosed for diabetes and takes both ibuprofen and paracetamol, there's 12 men and 6 women, but you've selected only these 6 women. So you know how much you will be going up if you were to say, or men, you would be going up by 12 people. So this is kind of intriguing because it gives you sort of a peek of the future without going there, and allows a little bit of a shortcut as you make progress in investigating things, into forming hypotheses, and into looking for insights in the browser. So something else I wanted to mention is you will see here there's this warning icon every now and then. This actually tells you that there's missing data. In this case, we know that there's one individual of these 6 women that is missing data in this field. And that's useful for you to understand how much missing data is there, because when you see that 5 of these 6 women have had cardiovascular, then you're like, oh, maybe it's not 100% prevalence of cardiovascular disease in this cohort. But in reality, the 6th person doesn't have any data in this field. So we don't actually know what diseases they had. They didn't answer the question. So it's useful for you to understand whether the distribution you see is because something looks like that, or there's a lot of missing people. And you can always apply an additional filter to exclude people with missing data. So if you don't want that 6th person, you can remove them from everywhere else. So that you say, I want only people whose non-cancer illness code is not null. Anyway, a lot of intriguing information here. You can always search here also for something like a disease and click and apply. There's almost way too many things for me to show you. But in reality, where I want to go with this is you can use this to generate your hypothesis, to look up something that you think might be happening in your cohort, or to even get smoking gun evidence for something. And unfortunately, this is synthetic data, so it's not meant for me to actually generate some meaningful insight. But if you look at the real UK Biobank data, for example, and you filter for diabetes, immediately you see men have twice as much as women, which is an interesting fact. Why is diabetes more prevalent in men? And that's kind of a known fact, but it remains a bit of a mystery as to why this is happening. So let me go ahead and remove some of these filters. And then we're going to go through some of the genome information. Okay, let me keep just the gender equals female. So down here this whole time, we had this variant browser. And this shows you the genomic variants that are part of your current cohort. This looks at that 2+ billion genotype matrix, and in real time filters for the variants that are part of these people in your cohort. At this point, I'm looking at the women, so we're looking at 27,000 people, and I'm looking at a specific region in the genome. So this is a bit of a genome browser of sorts. The location is chromosome 1, and I'm looking at a particular gene, PCSK9. This is a rendering of the gene structure, where the rectangles are the exons and the thin lines are the introns. And up here we see all the variants in my current cohort, which are visualized as these sort of lollipops where the height equals the frequency in the population of 50,000. So common variants will be looking like these. So this is an example of something with allele frequency 0.9, and rare variants will be like these. And in fact, in an exome experiment, you see that most of these variants overlap these exons because this is typical of an exome experiment, and they are rare. So as you add all the rare variants of these 27,000 people, they become this smudge here of all these exons that prove that most people have rare variants in these coding locations. And in fact, if you were to take a closer look from a genetics point of view at what the 50,000 first exomes that have been publicly made available look like, almost 80%, if not more, of these variants are singletons. Only one person has any given variant. So all of these variants appear down here also as a table: the location of the variant, DB/SNP RSID, what is the actual variant, and what is the worst consequence for every gene that it overlaps. The population allele frequency in the 50,000, together with an allele count, if you were to hover over, and then the same frequency in GNOMAD, which aggregates all the other databases. So it's an interesting proxy of has anyone else ever seen it and whatnot. Sometimes the GNOMAD frequency is 0, meaning that GNOMAD has enough coverage and has seen that location, but has never seen that particular allele. So there's never been an A in GNOMAD. That's what this 0 means. And sometimes this is empty, meaning that GNOMAD doesn't have a designation as to whether this variant has been discovered or not, because it hasn't had enough coverage or it's outside of GNOMAD's aggregation coordinates. So this is a table that you can quickly filter. So if you're interested, is there any woman here that actually has a stop gain mutation in PCSK9, you can apply a quick filter here to see that there exists all these mutations, and some are more frequent, some are less frequent in the population, and so on and so forth. So if you find some interesting mutation, you can always investigate. And there's a couple different ways that you might be using this. One is to say, I'm going to filter for my cohort of choice. So I want women with diabetes who take a specific medication. And as you apply this, you go down to a very small number of individuals. So if I refresh my dashboard here, you'll see I'm gonna go down to 3 people, really 3 women that have been diagnosed, have been hospitalized with a secondary code of diabetes and have taken this medication. So up here you see the pheno information for these 3 women, and down here you see their genome. So now that I'm down to 3, almost all variants will be common variants and will have high population allele frequencies. But if I think that there's a gene of choice that is implicated in that, let's say I believe CFTR causes this combination of diabetes with a particular drug, I can go to that location and then I can see, is there a rare variant that all these 3 women have? And it's not. Therefore, okay, my hypothesis that this disease is caused by this gene might not be right. You can also do the alternative. You can say, okay, let me keep the women as a phenotypic filter, but apply a genomic filter on top of it. So I want women who, for example, have any kind of high impact mutation in BRCA1. The genomic filter here allows you to say what kind of mutations you want someone to have in your cohort based on their functional consequences on any one of the genes that you enter here. So when I apply this, now we're going to be looking at a combination of geno and pheno criteria. They have to be women and they have to have any kind of damaging mutation in BRCA1. So again, the technology behind the scenes will scan the gigantic matrix for a combination of things that you say here and conclude, okay, there's 152 people that match the criteria. And these are all the diagnostic codes they have. In fact, if I undo this, you'll see, okay, what's the most prevalent disease? Oh, it's the circulatory system, and it could be hypertension. But here you see it's 25 out of 152, so maybe that's similar to the general population. So maybe changes in BRCA1 don't really cause hypertension. But when you do it with real data, and again, unfortunately, I can't show that to you today, but if you were to go to the UK Biobank 50,000 released exomes and look at, for example, people who have a damaging mutation in HBB that encodes for a part of hemoglobin, you'll notice that they are enriched for anemias. So these are the kinds of things that you can investigate very quickly, that research might have taken decades to discover painstakingly, but you can, in very few seconds, quickly interrogate. And alternatively, you can always go and pick a mutation of choice and say, okay, you know, I'm interested in this mutation. There's something very important about this mutation that I need to discover what it does. So you can say, instead of women with BRCA1, I want women with this exact mutation. And then you can look at exactly what are their diagnostic codes, what are the treatments, have they been diagnosed by a doctor, do they feel miserable, anything you might think is meaningful for you to investigate. What does that mutation do, and so on and so forth. Lastly, I'd like to discuss another feature here on the fact that you can actually click any variant and go to a page that discusses the variant in a bit more detail. And it is something that gets created out of the variant calling file data that gets provided by the UK Biobank. And so you see in this location, the T goes to a C. That's all that's been observed. And this is in 100,000 instead of 50,000 as an example. So these are how many people have T and T, how many people have T and C, how many people have C and C versus no call, etc. So it looks like it might or might not be in Hardy-Weinberg equilibrium. And these are all the genes that this variant is overlapping. These are all the consequences of every transcript and so on and so forth. But whenever a variant has multiple alleles, this becomes a little bit more complicated. In fact, if we were to reset these filters, let me refresh my dashboard, and let me go back to PCSK9. We can find some examples like this one where there are more than one allele in the same location. This is not so infrequent actually in exome or next generation sequencing results, unlike our array genotyping data like what's been previously published by the UK Biobank. Here you could have multiple choices at any given locus, and here that's a triallelic location where you could have a C or an A or a G in any combination, right? So there's that many people have C and C, C and A, C and G, A and A and G, and so on and so forth. And this becomes even more intriguing when you have an indel that is a low complexity array, for example, something like ACG, 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 that some people have 3 copies or many different copies. I don't think we have a very good example of that readily available, but this is something that's kind of difficult from a population point of view to take a look at. And the technology here allows you to look at some of these arrays and see exactly what copies exist in how many people in the population. Anyway, we're reaching the end of this presentation. I want to give people a lot of time to ask some questions. So let me stop here and give it back to Brenton to see where we can go from here.
Perfect. Thank you so much, George. As a reminder to everybody, you can submit your questions through the Q&A or the chat icons at the bottom of your Zoom window. Our first question is asking how the first 50K patients were selected from the whole population.
Right. So the UK Biobank as a whole has 500,000 participants. Those were selected with no specific criteria other than they have to be above 40 years old and live in certain, in the UK, right? And that's what's available in the UK Biobank. Then Regeneron, with the help of other pharmas, has started sequencing slowly those people. So the first 50,000 of the 500,000 have become publicly available. And that's what we're trying to showcase here. So it's a question of how did Regeneron choose those 50,000 first patients in their sort of priority of eventually sequencing everyone and making it publicly available. There's a paper that's been published by Regeneron and GSK on the criteria for why they chose these 50,000 people to work with first. So you can take a look at the paper. I think it's related to whether they had certain diseases or enriched for certain things or had imaging information and whatnot.
Great, thank you, George. We've had a couple questions that are kind of similar related. What are the requirements to access the UK Biobank data? And then as a follow-up of that, how do people gain access to the Cohort browser?
Requirements to access the UK Biobank data. So to access the UK Biobank data, you need to submit an application to the UK Biobank. There's a well-characterized procedure you can read on the UK Biobank website. You go through a process where you describe your credentials, who you are, what you do, and then submit a research proposal, what you want to investigate the UK Biobank data for. Then you choose which portion to work with, although you could potentially add a ton of fields in your cart to work with, for example, in the context of some big machine learning project that perhaps needs to look at different kinds of values and whatnot, and then submit that to the UK Biobank. They will review your application, they will receive some small payment, and then you will be allowed to retrieve your basket of sorts, whichever of the thousands of fields you've added to your cart. And if you've added the genomic information from the exome sequencing, you're allowed to access that as well. And then you can bring those to our platform and instantiate this browser together with all the other features that DNAnexus Apollo allows you to do. Sorry, what was the other question?
Access to the Cohort browser.
Yes, so sounds like a question for our sales team.
Yes, probably, I would agree there. So we can follow up with you after and answer that question for you. Someone was asking about the age range that was set for the data. Why was it set between 40 and 60?
Right, that's an interesting question. My take on this, and it's, I'm not, obviously that's perhaps a question for the biobank, but I understand that they wanted people who aren't that young, so that they've sort of gone through some diseases and there's information about what has happened to them. So if we look at the much younger population, they appear to be healthier because they haven't lived long enough for all of these things that show up later in life to be there and to perhaps be more meaningful in future health related research.
Perfect, thank you, George. Got a question asking if there's a way to view frequencies of variance in one cohort to other cohorts or the entire population.
It is not available in this version of the browser that I showed you. In the full product, there's a capability, once you build the cohort, to snapshot it and start computing some analyses, including computing frequencies in that cohort, as well as computing some more rigorous statistics that need precise sort of mathematical formulas as opposed to this. So there's a roadmap for this product that includes some visual tweaks that can help someone compare across two cohorts and see stacked distributions of something against something else. You can look at age in women versus age in men as distributions on top of one another, or whatever two cohorts might be from a visual standpoint. That's coming soon, but what's also already built in the product, and again, I'm not showing it today, is the capability to launch analytics on your cohort. So you can actually compare your cohorts for something like GWAS case control or gene enrichment or compute some more intricate formulas in R and Python and whatnot. Behind the scenes, this technology leverages big data analytics. So it doesn't matter whether you're working with something small or something large. You can save your cohort and then you can leverage our big data technologies to interrogate your cohort from any programming language, from Spark, from Jupyter Notebooks, from Pling, and so on and so forth. I'm afraid that could be a whole another chapter of its own, but hopefully that gives you enough ideas to keep your creative juices flowing.
Thank you, George. Next question is asking about the software used to call variants, and also asking what the depth of the whole exome sequencing.
Right. Great questions. Again, I think that's a question for Regeneron, as they're the ones that are in charge of the pipeline. I believe they're using GRCH38. They're doing mapping with BWA and variant calling with a couple of different pipelines. One uses a variant caller called WeCall, 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 and another that uses GATK. And then they conduct aggregation and joint genotyping of the VCFs into a project level VCF that has all the people as columns and all the variants as rows, using a technology that we've created called DNAnexus. That's just an R&D project of ours that allows you to do joint genotyping at scale beyond what current software allows you to do in an open source manner that's perhaps limited. In fact, ours is also open source. You can Google DNAnexus on GitHub for that particular step. The depth, I'm not so sure what exactly it is, but I believe they've mentioned that in the paper. So their paper is in bioRxiv and discusses some of these things, how they've done some of the analytics. So I would refer you to that.
This question's kind of a great follow-up to that, asking about a roadmap available for the UK Biobank. I believe in regards to rolling out of the rest of the data.
Okay, so if this is a question on the UK Biobank itself having a roadmap, they do indeed. Actually, if you go to their website, there's a page on future data availability, and it says that they're waiting for this consortium to finish sequencing of the exomes, and after their exclusivity expires, they will become public. So I think the first 50,000 became public now. The next 100,000 will become public maybe in 2020, and eventually all the 500,000 will become public shortly thereafter. Separately, the Biobank has announced an independent track to sequence the whole genomes of 500,000 people slowly as well through a completely different sort of parallel track. They're also adding GP record linking, some other biomarkers or other assays and whatnot. So I think they are continuing to conduct the imaging experiments and whatnot. So in terms of how the resource is growing, I'd like to refer you to the website so you can learn more about that. And they do have, the Biobank has scientific committees that are in charge of, what's the direction that the Biobank will go to, but they're obviously open to ideas from the community and collaborations. In fact, this project that Regeneron undertook was something I think that the Regeneron took initiative to suggest to the Biobank. In terms of our product roadmap, it's not quite based on UK Biobank. We have a lot of pharma customers, and we usually typically discuss with them, and they provide feedback on how they want the product to evolve. We announced last year that we have an initial collaboration co-development sort of agreement with MedImmune, where we did some of the stuff that you see here were done in consultation with them for their internal use and so on and so forth, to give you an example.
Perfect, thank you, George. A question asking about how long these cohorts will be followed up on. I believe like the cohort data from the study, if they'll be followed for life.
I think they're actually trying to do that to some extent. They are constantly asking the participants to come back for reassessments. They're trying to get information from their health records in a longitudinal manner, although the granularity of doing so is not real time. As I understand it, it happens maybe every few months or a couple times a year. I'm not so sure exactly what's the workflow for the biobank to do that, but it is indeed a evolving data set. In fact, if you as a researcher go and ask for access and you select your fields and you create your card and your application gets approved and then you download your big data set, you can go back later in the future and refresh, quote unquote, your card, and you'll get the updated values that, some things might have changed, more things might have been added, some fields that were measured on fewer people now are measured on more people, and so on and so forth.
Perfect, thank you. I actually have a question for you, George. Is this browser only available for UK Biobank data, or is it also available for other data?
It's not available only on UK Biobank data. It's just a showcase of our technology, which works pretty much with any data, any structured data that can be fed into the system, and any metadata that can be provided. So we have a technology where you give us the values that you're bringing in and some information as to what they are, title, units, link out, and so on and so forth, as well as your other data modalities, your genomic data, and what have you. And then we have an ingestion mechanism that transforms and loads all of that in our big data technology and instantiates these and other applications, and behind the scenes also databases where you can interrogate them with SQL and whatnot. So you can envision this for your clinical trial data or for other studies. And I apologize, by the way, if you hear ambulances or fire trucks in the background, but we're in San Francisco where this is a very frequent occurrence.
All right, perfect. Thank you so much, George. I think that'll be our time for today. Thank you everyone for your participation and for sticking around with us for this today. This was great. If we didn't get to your question, we will answer it in a follow-up email with you. We apologize for that. But if you do have any other questions that you'd like to ask, you can contact us at info@DNAnexus.com. Also, the recording will be going out probably early next week, so keep an eye on your inbox for that. Again, thanks again for joining us, and have a good rest of your day.
Thanks, everybody. Take care.